Welcome learners to the second episode of Crisp Review Biochemistry by Poonam Agarwal. And in this short video, I'll be discussing you about the HMP Shunt pathway, which is a very important pathway in the cell. And this pathway is occurring in the cytosol and the glucose is getting oxidized here. HMP Shunt pathway, the complete name is hexose monophosphate shunt pathway. And this is also known as pentose phosphate pathway because we get to have a number of pentose phosphates in this pathway. One thing important you must know, though we are having the glucose oxidation in this pathway generates no ATP. This pathway is not meant to produce ATP. ATP production is taking place in the glycolysis, but in HMP shunt pathway, ATP is not at all generated. The purpose of this pathway is the production of NADPH and ribose 5-phosphate. You know the importance of ribose 5-phosphate. It is needed for making of nucleotides. And all cells, they need nucleotides. So ribose 5-phosphate is actually coming from this HMP shunt pathway. And as far as this NADPH is concerned, this is needed for handling the oxidative stress in the cell. So oxidative stress is being handled, right? And for reductive biosynthesis means that we have reduction of intermediates, like for example the fatty acid synthesis. During fatty acid synthesis we have certain intermediates which need to be reduced with the help of NADPH. Cholesterol biosynthesis, bile acid biosynthesis, sterol biosynthesis, right? So those are the reductive biosynthetic pathways where the NADPH is supposed to supply the reducing equivalent, and this NADPH mainly comes from HMP shunt pathway. So now we're going to discuss this pathway which has got multiple steps in it, right? So I'm just going to tell you that we are not going to talk about single glucose residues getting utilized in this pathway. Rather, we are talking about 3-glucose 6-phosphate together. For convenience, like this pathway is a huge pathway, like you have so many reactions. And for convenience, you divide this whole pathway into oxidative phase and non-oxidative phase. You have oxidative phase reactions and you have non-oxidative phase reactions. Oxidative phase reactions are irreversible reactions. They're single direction reactions, and reversible reactions are seen in non-oxidative phase. And when we talk about NADPH production, it is getting generated in oxidative phase, and ribose 5-phosphate is being generated in non-oxidative phase. Now to start with this pathway, if you can see that we have the glucose 6-phosphate which is converted to 6-phosphogluconate, and the related enzyme is 6-phosphoglucose 6-phosphate dehydrogenase enzyme, which is G6PD, popularly known as G6PD enzyme. This 6-phosphogluconolactone is converted to 6-phosphogluconate via hydrolase enzyme, and 6-phosphogluconate is next converted to 3-keto-6-phosphogluconate by 6-phosphogluconate dehydrogenase, and this 3-keto-6-phosphogluconate is converted to ribulose 5-phosphate. So you understand we have two dehydrogenases, and both these two dehydrogenases, G6PD and 6-phosphogluconate dehydrogenase, both these dehydrogenases, they require NADP as a coenzyme. So what happens? NADPH is getting generated. NADPH is getting generated here, right? This is how, and these reactions are all irreversible reactions. Once the ribulose 5-phosphate is produced, what happens? We have epimerase enzyme, we have isomerase enzyme, and they're going to convert certain ribulose 5-phosphate to xylulose 5-phosphate and ribose 5-phosphate respectively. Next to that, there is a role of trans-ketolase enzyme, which is thiamine pyrophosphate dependent enzyme, and this enzyme is going to convert xylulose 5-phosphate and ribose 5-phosphate to glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. After that, transadolase is converting glyceraldehyde to fructose 6-phosphate and pseudoheptulose to erythrose 4-phosphate. Then this erythrose 4-phosphate is going to get acted upon by trans-ketolase once again, and then xylulose 5-phosphate will be acting as a second substrate here, and the product is going to be fructose 6-phosphate and glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate ultimately may enter in the glycolytic pathway. So in nutshell, if I would like to represent you the reaction of HMP shunt pathway, you'll be needing 3 glucose 6-phosphate along with 6 molecule of NADP plus, which is the coenzyme, and they're going to convert to 2 molecule of glucose 6-phosphate, right, and 3 carbon dioxide and 1 glyceraldehyde 3-phosphate and the simultaneous generation of 3 NADPH. Sorry, 6 NADPH will be seen. Simultaneous generation of 6 NADPH will be seen. Let me write it properly. You'll wonder that consumption of 1 glucose 6-phosphate in this pathway is going to leave you behind 6 molecule of NADPH. So it's a very rich source of NADPH. I can show you 6 NADPH. You can see 1 NADPH here, 2, 3, 4, 5 and 6 NADPH. For the loss of 1 glucose 6-phosphate, we are getting 6 NADPH in this pathway, right? In G6PD deficiency, G6PD is the rate limiting enzyme. So now coming to the next aspect of this pathway, and that is the G6PD deficiency. G6PD deficiency is making the person prone for hemolysis, because G6PD deficiency, the supply of NADPH is going to get affected. And this NADPH is important to handle the oxidative stress, as I have told you. So what happens? The oxidative stress is not being handled, and you see the H2O2 is going to damage the cell membrane. How the oxidative stress is being handled by NADPH? This is H2O2, which is generated in so many instances in electron transport chain due to incomplete reduction of oxygen. This H2O2 need to get converted to water. For this you need to have reduced glutathione, and this reduced glutathione will be converted to oxidized form. For maintaining the reduced glutathione supply, you need to have NADPH, right? So the role of NADPH is here to maintain the glutathione in the reduced form. This is glutathione reductase enzyme, and this is glutathione peroxidase enzyme. So if there is no supply of NADPH due to G6PD deficiency, reduced glutathione will not be there, and H2O2 will be increased so much. And this H2O2, by a non-enzymatic reaction, that's called Fenton reaction, will be converted to hydroxyl ion, which will damage the membrane. So that is what is seen in G6PD deficiency, that supply of NADPH is affected, and this results in hemolysis. Patient is prone for hemolysis. So one such case I have brought here, like a 23-year-old African boy is coming with a fever, chills and rigor. So malaria was diagnosed and patient was prescribed Primaquine. So on taking Primaquine, patient fever was gone, like malaria was treated, but after a few days patient presented with sign and symptoms of hemolytic jaundice, fatigue, breathlessness, pale skin, dark colored urine, and blood investigation when done was shown total bilirubin as 5.3 mg%. Hemoglobin was also low. Conjugated bilirubin was 3.3, unconjugated was 2 mg/dL. So this was the sort of picture of hemolytic jaundice. You can see the ectric sclera of this patient. So then such kind of presentation is very commonly seen on intake of certain strong antibiotics, Primaquine, certain food items like fava beans. Like for example, if you read here, norfloxacin, acetanilide, cotrimoxazole, dapsone, doxorubicin, furazolidone, methylene blue, and number of antibiotics, Primaquine, and all they are going to precipitate the oxidative stress in the cell. And fava beans is also which is commonly being used in Chinese food item, and excessive consumption of this fava beans also precipitates the hemolytic episode in the patient having underlying or borderline G6PD. Right, the chemicals known as vicin and convicin is responsible for hemolytic attack. They are found in fava beans. Right, so with this you understand that what is the significance of HMP shunt pathway in maintaining the equilibrium in the cell, right? And very quickly I will overview the uronic acid pathway as well, which is taking place in the liver, oxidizing glucose to number of intermediates. The most important intermediate is UDP glucuronic acid, which is important conjugating agent, And what you must know that in this particular pathway we have an enzyme called xylulose reductase, right? It's written here, or otherwise called xylitol dehydrogenase, and its deficiency will result in essential pentosuria. Deficiency of xylitol dehydrogenase, otherwise called L-xylulose reductase, results in excretion of L-xylulose in the urine, and this condition is a benign condition which is called essential pentosuria, right? And this pathway is not making ascorbic acid in human. Ascorbic acid or vitamin C is not synthesized in human due to lack of L-gulonolactone oxidase enzyme. This enzyme is actually there in the lower species, in the primates, but not in human. Vitamin C is not synthesized, not in human. It's not synthesized by this pathway, and the reason is lack of L-gulonolactone oxidase, which is important for making of vitamin C. So we are left with the importance of this pathway in making of UDP glucuronic acid, which is important for the biotransformation, conjugation reactions of various xenobiotics, and also for making of mucopolysaccharides, right? So this is brief regarding uronic acid pathway. So with this I complete this second episode of review of biochemistry. Thank you very much. For any query you can ask me in the comment section.